Our topic for this video is renal physiology. This will be a quick review for the clinical lab. Afterwards, we'll go over questions and answers based upon our MLS ACP Board of Certification study guide. First, a brief intro to renal anatomy. Now, let's take a closer look at the microanatomy and the important role of essential renal functions. In the renal cortex lies most of our glomeruli and Bowman's capsules. The majority of nephrons are cortical nephrons, named so because they lie in the renal cortex. Cortical nephrons are responsible for removal of waste products like urea and ammonia and reabsorption of nutrients such as glucose and amino acids. Near the renal medulla lies the juxtamedullary nephrons with longer loops of Henle. They have the primary function of concentrating the urine and maintaining water balance. The amazing ability of the kidney to clear waste products selectively while maintaining essential water and electrolyte balance is controlled by healthy nephrons that regulate renal blood flow. They travel through afferent arterioles to a tiny tuft of capillaries called the glomerulus. Glomerular filtration is a critical renal function, as well as tubular reabsorption and tubular secretion. The glomerulus is a tiny tuft of capillaries that filters renal blood flow. When healthy, it keeps cells and large molecules greater than 70,000 Daltons within the renal blood flow. The ultrafiltrate collects in Bowman's capsule, influenced by hydrostatic and oncotic pressures, controlled by hormones such as the renin, angiotensin, aldosterone system, also epinephrine. Here we see sodium chloride get filtrated, but the protein molecules are not, unless there is a glomerulonephritis, diabetic kidney, nephrotic syndrome, or other nephritic diseases. FYI, this is a light micrograph section of a normal glomerulus encased in Bowman's capsule. Let's take a closer look at the glomerular wall into Bowman's space. Here we see glomerular hydrostatic pressure filtering the blood through porous fenestrated endothelium, glomerular basement membrane, and slit diaphragms in between podocyte foot processes, forming the urine ultrafiltrate. Creatine phosphate is a breakdown product of food and muscle catabolism. Creatine is broken down into creatinine, a waste product which is excreted in the kidneys. Creatinine is valuable for the clinician to determine if an unknown body fluid is urine because it should be high in urine and absent or low in other body fluids. The average GFR is 120 mL per minute, depending upon factors such as age, sex, race, body surface area, and muscle mass. It is much lower in acute and chronic kidney injury, previously known as acute and chronic renal failure. Estimated GFR is by an equation, CKD EPI 2021, the chronic kidney disease epidemiology correlation formula using serum creatinine, age, and sex. Creatinine clearance is the volume of blood plasma that is cleared of creatinine per unit of time, measured in mils per minute. Although it is a bit of a hassle for the patient to collect 24-hour urine volumes and the lab to measure serum and urine creatinine together, it is a useful measure of approximating GFR. Both have limitations for muscle mass and race not taken into consideration. The creatinine clearance is calculated with urine creatinine concentration divided by serum creatinine concentration, milligrams per deciliter. Total urine volume is divided by 1440 minutes in 24 hours to get the volume per minute. Body surface area is calculated with a nomogram, plugging in the patient's height and weight to get the body surface area. Note that after doing the algebra, the unit dimensions cancel out, leaving mils per minute, which reflects the glomerular filtration rate. Renal reabsorption by active transport of glucose occurs at proximal convoluted tubules, but only up to the renal threshold at 160 to 180 milligrams per deciliter. High glucose levels above the renal threshold spills into the urine, forming glucosuria, seen in patients with diabetes mellitus. Glucose is osmotically active, pulling water molecules along with it, thereby causing an osmotic diuresis, where diabetic patients urinate frequently with high volume, known as polyuria. When they drink a lot to compensate for diuresis, it is known as polydipsia. Afferent arterioles supply each glomerulus with renal blood flow. To efferent arterioles, to the vasa recta, Latin for vessel, straight, lies within both renal cortex and medulla. The blood flow in these straight blood vessels establishes a countercurrent mechanism with the loop of Henle and serves to maintain an osmotic gradient for final concentration of the urine before draining into the collecting duct. Water evaluable solutes are filtered and reabsorbed in the proximal convoluted tubule and loop of Henle, where an osmotic gradient of the renal medulla allows passive reabsorption through aquaporin channels in the tubules to cross over to the vasa recta blood vessels to form a concentrated urine in the tubules. See the osmotic gradient go from 300 milliosmoles per kilogram to 1200 at the loop. This system is known as the counter current multiplier system. Normally, water will not go through impermeable ascending loops of Henle, distal convoluted tubules, and collecting ducts unless there is a disease or hormones. In a 24-hour period, a normal urine volume is 1 to 2 liters per day. And here's some terminology. What hormones can affect this total urine volume? Antidiuretic hormone opens aquaporin channels at the distal convoluted tubule and collecting duct, and I'll explain in detail soon. Antidiuretic hormone, also known as ADH and arginine vasopressin, is a hormone synthesized by the hypothalamus and secreted by the posterior pituitary gland. When we are dehydrated and we feel thirsty, the hypothalamus senses hypernatremia and hyperosmolality, and the posterior pituitary secretes ADH, which travels to the nephrons and opens up water channels at the distal convoluted tubules and collecting ducts to reabsorb and conserve precious water. The renin-angiotensin-aldosterone system helps regulate flow of blood to and within each glomeruli, responding to changes in blood pressure and plasma sodium levels that are monitored by the juxtaglomerular apparatus. The macula densa is part of the JG apparatus near the distal convoluted tubule. When the JG apparatus senses low blood pressure and plasma sodium, it triggers the macula densa to secrete the hormone renin. Renin enzymatically catalyzes the conversion of angiotensinogen to angiotensin 1, which goes to the lung alveoli. In the alveoli, angiotensin 1 is converted to angiotensin 2 by ACE, angiotensin converting enzyme. Angiotensin 2 corrects renal blood flow and sodium reabsorption, causing blood pressure to get back to normal. But wait, there is another trigger for another hormone. Angiotensin 2 stimulates the adrenal cortex in the adrenal gland to secrete aldosterone. Aldosterone is a mineralocorticoid hormone secreted by the adrenal cortex to act on the distal convoluted tubules and collecting ducts to conserve sodium. It stimulates sodium potassium exchange, leading to correction of low sodium to normal, thereby correcting the low blood pressure to normal. So far, we've covered three important renal functions. The fourth one is renal tubular secretion. What causes ammonia buildup? In summary, breakdown of proteins leads to amino acid catabolism and transamination in the liver, generating ammonia. Ammonia and ammonia ions are very neurotoxic to our brain cells, causing cerebral edema, coma, and encephalopathy. So, how does our body get rid of the neurotoxic ammonia? Ammonia is processed in the liver through the urea cycle and excreted in the urine, getting rid of nitrogenous waste. However, some ammonia is excreted by renal tubular secretion. The key enzyme in our blood to maintain pH homeostasis is carbonic anhydrase that catalyzes the conversion of CO2 to carbonic acid, which loses a hydrogen to leave bicarbonate, our main base buffer. Excess hydrogen ions in renal tubular cells are secreted in the tubular urine. Hydrogen ions combine with ammonia to form ammonium ions, which is excreted in urine. By the way, hydrogen ions also combine with hydrogen phosphate, forming dihydrogen phosphate, seen as the familiar turbid amorphous phosphates in urine. The body normally produces acids which are eliminated by the lungs and kidneys with blood carbonate serving as the main buffer to maintain homeostasis. Otherwise, we will see acidosis or alkalosis in these patients. When it is caused by the pulmonary problem, the patient gets respiratory acidosis from buildup of PCO2. When the patient hyperventilates, we see respiratory alkalosis. Likewise, the body may get metabolic acidosis or alkalosis if there is too much acid or bicarbonate. Do you remember the equation? The Henderson-Hasselbalch equation is a useful formula for estimating the pH of a buffer solution, such as blood, which is a crucial aspect of maintaining homeostasis in the human body. For blood pH, the equation is expressed as pH equals pKa plus the log of the base over the acid. The bicarbonate carbonic acid pair is the most important buffer pair in plasma with a normal ratio of 20 to 1. So log 20 is 1.3, making the normal pH 7.4. By tubular filtration, reabsorption, excretion, and secretion, renal compensation for acid base balance is one other way besides plasma bicarb to maintain pH homeostasis. Thank you for your time, attention, and support, and we hope to see you in the next video.